I'm not sure about you, but when I first learned about cellular respiration, it felt like I was in a foreign city that I'd never been to before, and I was getting directions to some obscure restaurant on the other side of town in a language that I didn't speak. So maybe you identify with me a little bit. It gets easier with some repetition, and just like getting those directions, sometimes people have to point and draw a map and use smaller words, and so expect to have to go through it a few times and sort of concentrate on what is that thing so that it makes sense to you as I give you directions. I'm going to try it one more time in a different approach, but I encourage you to go back and look at other videos, look at different people's videos, reread the book, look at diagrams. It takes a little bit to put it all together. Remember, the main point is to make ATP so that you can do work. This is a fairly simple overview of aerobic cellular respiration. And remember our basic formula: glucose is broken down in the presence of oxygen, yielding water and carbon dioxide. I'm not going to worry too much about most of the numbers. Our focus is really ATP because that's what we're after. We want to extract the energy from glucose and make ATP. One number I might mention is, since you should know that glucose is a 6-carbon sugar, then it should make sense that we have 6 carbon dioxides. But other than that, the numbers are not the main point. How much are we talking about? A lot of times we do talk about another number. For 1 glucose, we make about 36, give or take, ATP. It's an interesting theory, but like, how many glucose molecules do I take in? I did kind of a back of the envelope calculation and said, what if we had a teaspoon of glucose? And that's the number I came up with for the number of ATPs. So we'll probably take in more than that in terms of calories each day, so we're dealing with some pretty big numbers. Maybe it's a little easier to break it down into a single glucose. I'm going to keep score as we move along here. ATP, I'm looking for 36 of these, and these 2 electron carriers also are helpful in the end in making ATP. So I'm going to track them as well. This is basically my energy scorecard. So let's break things down as the following. So glycolysis is part of what I'm going to do, and then the Krebs cycle, I'm just going to shorthand those, and then we have a separate one, the electron transport system, and I'm going to add into this, I'll just put it in parentheses, that also includes oxidative phosphorylation. So there's a lot of stuff that goes on. The electrons move through and make ATP. Technically that's part of oxidative phosphorylation. Big term, but just generally we'll refer to this as the electron transport system or the electron transport chain. All right, so let's take this one step at a time. So starting with glycolysis, I have glucose, and so now I've taken care of my glucose. That's the only thing so far. I'm going to represent that like I have before with this 6-carbon sugar model. Glycolysis is a series of about 10 steps, and what I'm going to end up with is 2-pyruvate. So we're going to break that in half. That's going to release energy, and I end up with 2 molecules that are 3 carbons each. All right. Now I'm pretty much done with breaking that down. Don't have anything else from my equation, and these guys get passed into the mitochondria to start the Krebs cycle. But let's take a bit of an accounting here in terms of where we're at. Well, in this, I'm going to extract some energy in a separate set of reactions and make 2 ATPs. All right, technically I'm going to put in 2 ATPs worth of energy and get out 4, but I get 2 ATPs net from this. I also get a little bit of NADH from this, and just to be complete, I know that I'm going to give off some heat energy according to the laws of thermodynamics, and so there was more glucose, energy in glucose than I got out of the pyruvate, ATP, and NADH. So some of it had to have left as heat, just to be technical there. Going on to my scorecard here, then out of glycolysis, I got 2 ATP and I got 2 NADH. I didn't note that there, but that's how many I got, and I'll use those to make more ATP later on. And that's about it. So I'm pretty far from my goal of 36 ATP. So I'm going to switch colors so I can color code this a little bit and go on to the Krebs cycle. So I start that one with those same 2 pyruvate molecules, and I'm going to go through that step of acetyl-CoA conversion, and then we're going to go into this whole series of reactions that start and end with the same molecule. During this time, during this acetyl-CoA conversion, one of those carbons is given off, and as this other one makes it around, they're chopped up as well. Same thing with this one in that step, and then as it moves around this cycle of reactions, then this one breaks down too. And so a lot of what is given off is carbon dioxide. Check. There we go. So I can take care of that one. Obviously, in breaking down that molecule, that should give off some energy as well. I'm not going to do this in order. I'm going to show it a slightly different way. And so during this step, we're going to give off a total of 8 that should be DH molecules. 2 from this step and 6 from this one. That's for both of the pyruvates together. The accounting is not incredibly important, but it does give us a sense of where is all this energy coming from, and that is an important concept. So I got all those guys. I also have 2 FADH2 molecules. This is that foreign sounding stuff, right? These are just electron carriers, so they're carrying energy and they're going to carry it over here. And then I'm going to give off also 2 ATPs. So back to my scorecard, I'm up 2 more ATPs. Let me underline that since we can't see the difference. I also got a boatload of these guys. I got 8 of those, and I got 2 of those. And so all these came from the Krebs cycle. Pretty good energy payoff here. Still don't have a lot of ATP, but I do have a lot of energy left, because all these are, remember, my energy scorecard. I have a lot of energy left with these guys, and that's what's going to be used to make the remaining ATP in the electron transport system. The electron transport system, remember, occurs in the mitochondria, specifically in these membranes, this inner membrane here. And so I'm going to represent that with this membrane, and there's lots of proteins in that membrane. So this is the electron transport system. I'll represent these proteins in the phospholipid bilayer like this, and then we also have that ATP synthase molecule that obviously synthesizes ATP, so that's another really important one. So this is really 2 steps here. So I'm going to switch to red here and note that we're going to get this NADH, the NADH here, the FADH2, and they're all going to drop off those electrons into this electron transport system of proteins. And as those electrons move through, they are going to power the pumping of hydrogen ions across. Go through this one, and more flow across. And now I'm essentially done. And so all I've gotten is a bunch of hydrogen ions on the other side of a membrane, which if you remember back to our membrane transport, I said that is stored energy because all those hydrogen ions are trapped in here and they want back in. But the only way we're going to let them in is not through this membrane, but through this. And as those hydrogen ions come through, we're going to use that to make ATP. And obviously we're making that out of ADP and a phosphate. We're just putting the phosphate onto here. We need energy to do that, and there we have it. And this makes roughly 32. So on my energy scorecard, now I have my 36, and I've actually used these up to make that one. So again, the big point of this is electron transport system, really important because most of our ATP comes from there. These two come from the mitochondria as well. We only get 2 from the cytosol where glycolysis happens, right? And all these NADHs come from those first 2 sets of reactions that power this ATP. Let's see, are we done? Well, we made our ATP, and so that was the oxidative phosphorylation, just so you note. That's this guy right here. But it does say oxidative, and going back up to this formula, I've not dealt with either of these two. I've got ATP all cinched up, but not these guys. So this is where they're going to come in, oxygen and water. So I have this electron sitting here, and I can't just let it go out into the cell. That's a reactive oxygen species, or you might know it as a free radical. It could really cause damage. And so oxygen, molecular oxygen that we breathe in, is really good at accepting those electrons. And when it does, it can combine with some of the hydrogen ions in solution to become water. And so water is made in here. It's actually called metabolic water. About 10% of the water you need a day you actually make by breaking down your food rather than by drinking it in. It's kind of an interesting thing. So that's probably the hardest one to understand out of all of them. And 2 steps, hydrogen ion gradient created by electrons and their power. And then step 2, use that gradient to make ATP. Then we got to do something with the electrons, and that's where that comes in. And obviously then, if we don't have oxygen, then we can't accept electrons. If we can't accept electrons, then this whole system shuts down, and anything that produces these electrons also is in trouble as well. So that's why we breathe in and out all the time, and the more ATP we need, the more oxygen we need as well. So there's an overview. I hope that put a few things into perspective.